Regulation of gene expression in eukaryotes. The regulation of gene expression in eukaryotes is essential for responding to signals either from the internal or external environment. For cell specialization in multicellular organisms, which are made of different cell types, to perform its function, each cell must maintain a specific program of gene. Typical human cell expresses about 20% of its protein-coding enzymes protein-coding genes at any given time. The subset of genes expressed in each cell type is unique, allowing the cell to perform its function. Gene expression is sometimes equated with transcription, although the regulation of gene expression in eukaryotes can occur at many additional stages. The regulation of chromatin structure will occur in the two types of the chromatin in the eukaryotic cell, the euchromatin and the heterochromatin. The location of the genes promoter relative to both the nucleosome and the sites where the DNA attaches to the chromosome scaffold can affect whether the gene is transcribed or not. The euchromatin and the genes in the euchromatin are usually expressed because they are loosely packed. The genes within the heterochromatin are highly condensed and are usually not expressed. There are some chemical modifications to both the histone proteins and to the DNA of the chromatin. The histone tails are accessible to enzymes that catalyze the addition or removal of certain chemical groups, such as methyl, acetyl, or phosphate. Generally, the histone acetylation leads to opening up the chromatin and promote transcription. Histone methylation do the reverse, reduce the transcription and increase the condensation of the chromatin. Different sets of enzymes can methylate certain bases, not the histone protein, but a different set of enzymes can methylate certain bases in the DNA, usually in eukaryotes, the cytosine. DNA methylation leads to inactivation of transcription, as found in the inactivated mammalian X chromosome and in genomic imprinting. This is a question. Does the DNA methylation pattern transmitted after cell division? Yes. Enzymes methylate the daughter strand. Enzymes methylate the daughter strand after each round of DNA replication. Can the modifications of the chromatin be reversed? Yes, it can be reversed, unlike the DNA mutation. For example, the DNA methylation pattern can be erased and re-established during gamete formation. The epigenetic variations can explain why an identical twin acquire a genetic disease while the other does not, despite their identical genomes. This is a typical eukaryotic gene and messenger RNA, primary messenger RNA and the mature messenger RNA. There are here three upstream distal control elements, two proximal control elements, the promoter and the exons and introns. This is the DNA. Then it will be transcribed to the pre-messenger RNA containing introns, and then it will be processed into the mature messenger RNA containing the 5' cap. The introns will be excised, and the mature messenger RNA will contain a 5' untranslated region and a 3' untranslated regions. Both are important for many functions, including ribosome binding. And finally, the 3' polyadenine tail. How chromatin modifying enzymes control the gene expression? By making regions of DNA more or less accessible to bind to the transcriptional machinery. This transcriptional machinery is the RNA polymerase II transcription factors, either general or specific, and mediator proteins. Associated with most eukaryotic genes are multiple control elements. It means control segments. These are segments of non-coding DNA that serve as binding sites for transcription factors. There are two types of transcription factors, the general and the specific. The general is essential for the transcription of all protein-coding genes. A few of them will bind to DNA sequences such as databox, but the most will bind to other transcription factors and RNA polymerase. The specific transcription factors bind to proximal and distal control elements, and these are activators and specific activator and repressors of each gene. A given gene may have multiple enhancers. Each enhancer is active at a different time or in a different cell type. Each enhancer is associated with only that gene and no other. Enhancers can be thousands of nucleotides upstream or downstream of a gene or within an intron. The protein-mediated binding of the DNA is thought to bring the activators bound to enhancers into contact with the mediator proteins that interact with the proteins at the promoter. Some transcription factors act as repressors by binding to the control elements and blocking the binding of the activator, or by binding directly to the activator itself, so that the activator cannot bind the DNA. Some transcription factors affect transcription indirectly by affecting the chromatin structure. Some transcription factors recruit chromatin-modifying proteins that can, for example, acetylate or deacetylate exons near the promoters of specific genes. There are two types of structural domains in the transcription factor: the DNA binding domain and the activation domain. The activation domain binds to other regulatory proteins, while the DNA binding domain binds to DNA. This is a model for the action of enhancers and the transcription activators. These yellow spheres and these yellow lines constitute the distal control elements. These are 3 distal control elements, and these 3 together constitute the enhancer. The activator proteins, these yellow spheres, bind to the distal control element, grouped as an enhancer, and this enhancer, as I said, has 3 binding sites. Each one is called a distal control element. A protein-binding protein will fold the DNA so that the activators will come into contact with the RNA polymerase II and mediator proteins. A DNA-binding protein brings the bound activators close to the promoter. And this constitutes the transcription initiation complex. The activators, together with the RNA polymerase II and the general transcription factors and the mediator proteins, all of them constitute the active transcription initiation. In eukaryotes, the control of transcription depends largely on the binding of activators or repressors to the DNA control element. The number of completely different sequences in the control element is surprisingly small. Short nucleotide sequences appear again and again in the control element. Each enhancer is composed of about 10 control elements, and each bind to only 1 or 2 specific transcription factors. It is the particular combination of these control elements in an enhancer that is important in transcription regulation, rather than the presence of a unique sequence of control elements. The combination is the important factor. There is a very large number of possible combinations of control elements, and these control elements can activate the transcription only when the specific activator proteins are bound. These activators can be available at a certain time of development or in a particular cell type. This is an example. 2 cell types, liver cell nucleus and lens cell nucleus. Both of them contain identical genes. The liver cell nucleus and the lens cell nucleus both contain the genes that encodes albumin and crystalline. Albumin is a protein that is expressed mainly in the liver cell, and crystalline is a protein that is expressed mainly in the lens cell. So both cells contain the same genes for these two proteins. Each gene has an enhancer made up of 3 different control elements. The albumin has 3 control elements, this one green and blue and yellow, while in the lens cell nucleus, the crystalline gene is under the control of 3 other control elements, the green one and the purple and the orange one. Although the green control element exists in both the albumin gene and the crystalline gene control elements, but the combination of the control elements and their activators are different between them. So, although both enhancers have a green control element, but each enhancer has a unique combination of elements. All the transcription factors, activators, that are required for a high-level expression of albumin gene is present only in the liver cells, and all the activators required for a high-level expression of crystalline are present only in lens cells. The coordinated controlled genes in eukaryotes. In bacteria, there was an operons that contain a clustered genes. For example, genes that are associated with a particular metabolic pathway are clustered together and are under the control of only one promoter and operator, and these 3 regions constitute the operon. Actually, the operons are not found in eukaryotes. So, the case in eukaryotes is that the co-expressed genes in eukaryotes, such as genes including the enzymes of a certain metabolic pathway, are scattered on different chromosomes. The coordinated gene expression depends on the association of a specific combination of control elements with every gene of a dispersed group. The presence of activators that recognize these control elements and bind to them promotes the simultaneous expression of those. The coordinated control of dispersed genes in eukaryotes occur in response to certain signals. For example, a steroid hormone enters the cells and binds to an intracellular receptor, forming a hormone receptor complex, which functions as a transcription factor and enters the nucleus. Every gene whose transcription is stimulated by a certain steroid hormone has a control element recognized by that hormone receptor complex. Other signaling molecules such as growth factors, non-steroid hormones bind to the cell surface receptors and induce a signal transduction pathway that leads to the activation of a certain transcription activators or groups. Genes with the same sets of control elements are activated by the same signals. Now we will talk about the post-transcription regulation. The expression of protein coding genes is measured by the amount of the functional protein that they make. There are 2 titles that we will discuss regarding the post-transcription regulation. The first one is RNA processing, RNA splicing, the excising of introns and removing introns and ligating or joining the exons together. This is called RNA splicing, which different RNA molecules are produced from the same primary messenger RNA transcript, depending on which RNA segments are treated as exons and which are treated as introns. Regulatory proteins specific to each cell type will control the intron-exon choices by binding to regulatory sequences within the primary transcript. Alternative splicing increases the number of proteins that can be made from a single gene. Number 2, regarding the post-transcription regulation, is the regulation of translation initiation. For some messenger RNA, the initiation of translation can be blocked by regulatory proteins that bind to either the 3' or the 5' untranslated regions. Both regions are important for the binding of ribosomes. Number 3, the lifespan of the messenger RNA. The sequences that affect the lifespan of the messenger RNA are often found in the 3' untranslated region. For example, the messenger RNA in bacteria, the lifespan of it is just few minutes. And this is one reason how a bacterial cell responds rapidly to environmental changes. In contrast, the messenger RNA of the eukaryotes can survive for hours or days or even weeks for some. For example, the messenger RNA of hemoglobin in developing red blood cell are unusually stable. Now we will talk about the post-translation regulation. Number 1, protein processing and degradation. Eukaryotic polypeptides must be processed to yield the functional protein. For example, cleavage of the initial insulin to generate the active insulin. Some regulatory proteins are commonly activated or inactivated by the reversible addition of phosphate groups. Proteins destined for the cell surface will acquire sugars. It will constitute a glycoprotein. Proteins must be transported to their sites of action in the cell. Regulation can occur at the steps of modifying or transporting a protein. The length of time each protein functions in the cell is regulated by a selective degradation. To mark a protein for degradation, the cell attaches molecules of a small protein called ubiquitin to the protein, and proteasomes then recognize the ubiquitin tagged proteins and degrade. Number 2, the non-coding RNA that control gene expression. As we know, the protein-coding DNA accounts only for 1.5% of the human genome, and in addition to a very small fraction that constitute the genes for ribosomal and transfer RNA. The remaining 75% of the remaining DNA is transcribed at some point in any given cell. Introns account for only a fraction of this transcribed non-translated RNA. A significant amount of the genome may be transcribed into non-coding RNA, which plays crucial roles in regulating gene expression, either by regulating the messenger RNA translation or regulating protein structure. The non-coding RNA have two types. We will discuss only two types of the non-coding RNA. The first one is microRNA. It is a small single-stranded RNA that form a complex with one or more proteins. MicroRNA allow this complex to bind to specific messenger RNA with at least 7 or 8 nucleotides of complementary sequence. The microRNA protein complex binds to the 3' untranslated region of the target messenger RNA, and the microRNA protein complex then degrade this target messenger RNA or block its translation. If the microRNA and the messenger RNA bases are completely complementary all along their length, the messenger RNA will be degraded, and if the match is less complete, translation will be blocked either by inhibiting the binding of translation initiation factors or by ribosome stalling. A single microRNA can participate in regulating as many as several hundred different messenger RNAs, and about 1/3 of all human genes are regulated by microRNA. The second type of the microRNA, the second type of the non-coding RNA is the silencing RNA or the small interfering RNA. It is thought to arise as a normal defense mechanism as a result of virus infection. If the virus has an RNA genome, but it is not a retrovirus, the perfectly matched double-stranded RNA molecules that arise as intermediates in the viral gene replication, these double-stranded RNA are cleaved by a protein called dicer to give the silencing RNA. These silencing RNAs or siRNAs are targeted to the viral RNA the viral RNA will be the target of these siRNA, so that viral RNA will be cleaved as that indicated in the microRNA process. This process is particularly effective in plants. In addition to regulating the messenger RNA or controlling the messenger RNA translation, the silencing RNA can interfere with gene expression at the level of affecting the chromatin structure. For example, in some yeasts, silencing RNA interacts with other non-coding RNAs and with chromatin modifying enzymes to remodel the chromatin structure at the centromere, leading to the formation of a heterochromatin at the centromere. This is what we have for this topic, and I hope this was helpful, and thank you.